So it's just like the first chapter, Foundations of Biochemistry. It's like the introduction. You notice, this is called the Leninger book. Why did they do that? It's Albert Leninger. He's the guy who describes the citric acid cycle that occurs in mitochondria. Mechanisms for oxidative phosphorylation. Do you guys know what? I'm sure you guys have had to memorize the TCA cycle or the Krebs cycle and so forth. No, micro or core biology. Yeah. What's oxidative phosphorylation? So that occurs in the mitochondria where you have that proton gradient across the inner mitochondrial membrane, and then you create that gradient by transferring electrons down their, reductive potential, and then ultimately they end up at oxygen, and you reduce oxygen to water, and you generated a gradient of protons, and then that gradient of protons drives the phosphorylation of ADP to ATP, and that's what's known as oxidative phosphorylation. And we'll learn about that much later in the semester. And this is all occurs in the mitochondria, except for bioenergetics. Bioenergetics is just using like chemical energy in life, basically. So and so forth. So that's Leninger, and this is his picture, and they wants to show you that. The book's named after him. He's like a big guy in biochemistry. So learning goals for this are going to be distinguishing features of living organisms. Okay, and the structure and functions of parts of the cell, basically. You guys know structure and functions of different parts of the cell? Like, what's the nucleus in a eukaryotic cell do? Well, it's where you store all the information to make all your other compounds. That's where DNA is stored. And you get DNA and RNA metabolism, so that's where you produce your RNA molecules. You know different types of RNA molecules? Or do you know the difference between DNA and RNA? Anybody? What?
RNA is used for messages being sent.
That's one form of RNA. RNA actually can be used as the template also in viruses. There are examples of that. Retroviruses. So HIV virus is a retrovirus. There's other ones. But chemically, do you guys know what the difference is? What does DNA stand for? And RNA, you know? Yeah, deoxyribonucleic acid. What do you think RNA stands for? Ribonucleic acid. So the only difference chemically is deoxy, you're minus one oxygen between the two. So, and then we have energy transformations in a living cell. So, of course, we have lots of energy transformation. I'm going to talk a little bit more about that. Regulation of metabolism and catalysis. Did I say that right? Metabolism and catalysis. You'll see my words get jumbled because I'm bad at words. So don't mind me for the rest of the semester. Just laugh at me if I'm a fool. Okay? Coding of genetic information in the DNA, and I just said that, and then I also said you can use RNA as the genetic information also. Role of mutations and selection in evolution. For example, RNA is used as genetic information, but one of the problems with RNA being used as genetic information is you have more mutations. It's more subject to degradation. It's not as stable as the DNA. So that's why, for example, HIV can mutate so fast and become drug resistant. Biochemistry is the chemistry of living matter. It's a high degree of complexation and organization. So, in extraction, transformation, and systematic use of energy to create and maintain structures to do this work. So, if it's highly complex and organized, do you guys remember thermodynamics in general? Remember Gibbs free energy? That equation? So, what word do you think of in terms of thermodynamics when you think of highly organized and highly complex? What's the term thermodynamics that you would use for things that are really ordered? Or to put a number on the degree of order? equals, what is it? H minus TS or something. And really, this equation, typically you're using the change, so there's delta there, right? You guys remember what delta H is? What's that known as? Is it enthalpy? Remember that? And then delta S is what? You remember? It sounds a lot like enthalpy. Entropy, right. So entropy is the measure of disorder, right? So if you look at this equation, remember, normally things are spontaneous, right? When your free energy is negative, you'll have a spontaneous reaction. You guys remember that? So if your enthalpy is negative, that's favored, right? Where you're losing energy to the system, you have a negative enthalpy. But entropy also, if you have a positive, because you see I have a minus here, if you have a positive change in entropy, that means increased disorder. That's definitely favored. But because living systems are so highly organized, that means this is often, negative also, right? You have a negative change in entropy because we're trying to build more complex molecules. Those long DNA molecules, they're big molecules that are highly ordered, and same thing with proteins. They're polymers of amino acids that are highly ordered and structured. It's entropy. It's I can't know how to see. Here's my word problem again. It's entropically, I don't even know if that's a word, not favored to build like such a large ordered molecule, right? So that is going to take energy, and where do we get that energy from? From our, but we produce the ATP from the sugar we eat. If we were plants, it would be coming from that big thermonuclear reactor in the sky, or if we're like bugs down in the thermal vents, it'd be from the heat generated from Earth, right? So it takes a lot of energy to maintain this organization, and that's what we're going to be learning about in biochemistry. So interactions of individual components are dynamic and coordinated. You'll see it's all coordinated, everything's regulated. So we can make glucose in our body, and at the same time we burn it. But we don't want to do this two at the same time, right? We want to make glucose, or gluconeogenesis is what that's called, when we have an excess of energy, we have a surplus. We make glucose to store it. We want to burn when we have a deficit of ATP, when we need to make other molecules, right? Things like that. So ability to sense and respond to changes in the surroundings, and a capacity for fairly precise self-replication while allowing enough change for evolution. That's what we'll learn a lot about the DNA and how that we self-replicate that way, and then we do get changes, because DNA itself is just a chemical. Chemicals degrade spontaneously over time. Same thing with DNA. You cannot help but get mutations over time. That's typically how we end up getting cancer, eventually, in our somatics. So, picture from the book showing you the complexity and organization of the cell. It's very compact and tight with all kinds of different things. And we must take in nutrients, because it takes a lot of energy to maintain order. When we die, that's what happens. Everything goes to disorder, and we become one with the earth again. Right? And we can replicate each other, ourselves, basically. So the cell is the universal building block. This is like old news to you guys, I'm sure. You did this in core biology, probably high school biology. We know all of this. This is just old time review stuff. But all cells are going to share some common features. This is a bacterial cell and an animal cell. Well, something that's different between the two. It's kind of obvious. These bacteria have a nucleoid, whereas the animal cells or eukaryotic cells are going to have a nucleus. That's where the genetic material is stored. And eukaryotes like us, it's separated from the rest of the cytoplasm. Whereas bacteria, it's kind of there just in the center of the cell, not really physically separated by a membrane. They both share cytoplasm. These look like ribosomes. You guys know what ribosomes do? What are ribosomes for? Exactly. So they're the structures that make proteins. Do you know how they make proteins? They catalyze peptide bonds, but this is an interesting catalysis because normally when we think of catalysis, we think proteins do the catalysis. They're amino acids that do that catalysis. In ribosomes, it's actually the RNA that serves the catalytic function. So there's some proteins in ribosomes, but it can still catalyze peptide bonds if you strip those proteins out. Anyways, that's just a little interest. We'll see more. And those are called ribozymes or things that do that. Three distinct domains of life defined by cellular and molecular differences. We have bacteria, archaea, and then eukaryote. You guys know what these are? You guys have ever seen these before? These trees? Phylogenetic trees. So basically, you can trace back, these are like common ancestors where these groups split off. You can trace them back. You can develop these just with DNA sequences or amino acid sequences of particular proteins to see where different organisms split apart. And you can actually retrace and theorize and hypothesize what old ancient enzymes look like. And there's actually one of the biology teachers is doing that on the main campus, looking at alcohol dehydrogenase and looking at where in evolution we developed the ability to consume alcohol, and you ask questions whether we're consuming it just to get high or are we doing it for energy, to use the energy from the alcohol, because it is an energy source. 6 kingdoms. So I'm not going to make you memorize these kingdoms. I hate memorizing things, but yeah, there's a lot of memorization in this class. Bacterial cell structure. So I might ask you some questions on a test about different organelles or cell structures. This is just basic bacteria. You have they contain cell membrane, right? Pili, which can act as adhesive forces. A nucleolide is where the DNA or the nucleic acids are contained, and so forth. Ribosomes, we already talked about making proteins. This is some components. Cell wall created there is made out of peptidoglycans. You guys have had micro before, probably. Cell membranes is lipid and protein, and that produces a permeable barrier so you can keep, separate metabolism from the outside world and separate, be able to separate things. Nucleoid is DNA and protein. That serves, genetic information. Ribosomes. It's made up of RNA and protein that produces the proteins. The pili are protein for adhesion to other bacteria and conjugation, where they share some genetic information. Flagella is for motility, and cytoplasm is the aqueous solution where you get the metabolism to occur. It's where you make your proteins, you burn energy, produce ATP, use that ATP to make proteins, and copy DNA and so on. So eukaryotic cells are more complex. They have a nucleus by definition, and if you guys remember, that nucleus is separated from the cytoplasm via the nuclear membrane, right? And it does this to protect the DNA, and it's a site for DNA metabolism itself. And you have selective import and export via the nuclear membrane pores. Some cells are anuclear, like our red blood cells, so they're not used to produce more red blood cells. Red blood cells are just used to what? Deliver oxygen around the body. Well, hopefully. So they have membrane enclosed organelles. Mitochondria are used for energy in animals, plants, and fungi. Chloroplasts, of course, are energy in plants. And then the lysosome is for the digestion of unneeded molecules. Spatial separation of energy yielding and energy consuming reactions will help maintain that homeostasis. What does homeostasis mean? Anybody tell me? It's like keeping that same level. Things like to be steady until there's some kind of change in the environment that'll alter the homeostasis until you reach a new level of homeostasis. The cell's going to have to adjust to that change. But normally, cell wants everything to be the same. So, and you don't want your energy yielding to be right next to your energy consuming reactions. You end up with fuel cycles, like making brand new sugar and at the same time burning sugar. If you do that at the same time. That's known as a fuel cycle, and it actually uses more energy than it produces. So this is just a shot of animal cell, or bacterial, animal, and plant cells are different. So this is eukaryotic cells. You have a nuclear envelope that surrounds the nucleus, and then you have things like mitochondria, where we produce our energy by oxidizing fuels. The rough endoplasmic reticulum, or the rough ER, is a lipid layer, like lipid vesicles that are coated with ribosomes. And that's where you get proteins sorted and modified once they're produced, and so forth. So I might ask you a question about some of the different intracellular components of these. Most of it should just be reviewed for basic biology, right? And this is a plant cell. Difference in plant cells is they have what's on the outside, probably right here, cell wall, and so forth. And then they also have chloroplast, so they can create or capture energy from the sun. So cytoplasm is highly viscous solution because it's really crowded with lots of proteins and other molecules, salts and things. That's where you get the reactions to take place, right? So it's an aqueous environment. We know lots of chemical reactions like that occur in aqueous environments. Cytoskeleton itself, it consists of microtubules, actin filaments, and intermediate filaments, and they give the cell shape, help to organize the cell, and you can have intracellular transport paths along those microtubules and help the cell move. And this is just some histology that they show you. Like the red filaments are actin, and the green filaments are tubular, or microtubules, which are actually polymer tubules. And then in this case, the red is keratin, and I believe the green is again tubule. Yeah, the microtubules. And then you have our chromosomes with the centriomeres, and this is during, cell division. And another figure from your book, just to illustrate how crowded the cytoplasm really is. It's not just a body full of aqueous solution. You have tons of proteins and ribosomes and other molecules in the cytoplasm. So the basis of all life is the chemical reactions that takes place within the cell. And again, we talked about how complex and organized it is, and that organization complexity is really takes away from the entropy of that system. So we have to extract, transform, and systematically use energy to maintain that complexity. The interactions of individual components to be dynamic and coordinated, and the ability to sense and respond to changes in the surroundings. So that is where the chemistry comes from. And a capacity for fairly precise self-replication while allowing enough change. This is basically the same slide. Is it the exact same? I think so too. Sorry. So you guys have seen this before, right? It can be categorized based on our energy sources. So we have we get our energy from chemicals. A lot of plants and other, algae and things like that get it from light. So we're chemotrophs, right? Things that get them from there. So organisms that get energy from light are phototrophs. They get energy from light. A carbon source, they can be from carbon dioxide if they're getting the chemical energy, like from the vents or like sulfur and things like that. There can be chemoautotrophs, where they convert carbon dioxide through sugar through other chemical reactions. Hydrogen, sulfur, iron, nitrogen, carbon monoxide. They all use that as their energy source and can create sugar from carbon dioxide, just like the plants create sugar from carbon dioxide using energy from light rather than from chemicals. We're, on the other hand, we get our energy from organic compounds like sugar and fats. And And so we're chemoheterotrophs, and we use oxygen as our final electron acceptor. In all animals, this is all of us. If you're not using oxygen as the final electron acceptor, you can ferment things like yeast. You guys like beer or wine? Or yogurt even uses fermentation. Or inorganic compounds such as pseudomonas denitrogen. For example, they'll use nitrogen. Things like that. And this is just living systems. We extract energy. All in all, for the most part, Earth living systems, we get most of our energy from the sun. There are organisms that extract it from geothermal energy sources, but for the most part, our energy is coming from the sun. We just extract it from the food we eat. It had previously, for the most part, come from the sun. So we have this cycle where the photosynthetic cells capture the energy, produce chemical energy that we then consume as heterotrophic cells, and then release carbon dioxide and water again, and they turn that into oxygen and sugar or fat. So what's the molecular logic of life? So we look at chemistry that's behind it. You guys know what proteins do, what their purpose is for the most part? They're catalysts. For the most part, not all of them are catalysts, but they catalyze chemical reactions. What does that do? What's the point of catalyzing chemical reactions? Yeah, lowers the activation energy. So it speeds up that reaction. A lot of reactions might be spontaneous that occur, but they might take forever for one molecule to have that activation energy for that to occur spontaneously. So they're really, really slow. So organization of metabolism and signaling and storage and transfer of information. So this we do via our nucleic acids. And these are the ABCs of life, meaning we have amino acids, and these are all incorporated into our proteins. And the amino acid sequences of the proteins are what's going to give them structure and their function. These amino acids are then coded for by the nucleic acids that we use as their genetic information. And I'm sure you guys have learned about the 3-letter codes, how these nucleic acid sequences are used to produce amino acid sequences. And then we have different lipids. So these are the 3 main molecules that we have in the body. Amino acids used to make proteins, nucleic acids used to make RNA and DNA. And here's the main difference between the RNA and DNA is the sugar backbone. RNA has all its hydroxyl groups. DNA is missing the number 2 hydroxyl on that ribose. And then we have our lipids, fatty acids. And you can say that there's like a molecular hierarchy towards life. So we start off with these small molecules. They come together, the sugars will come together to produce structural molecules like cellulose for plants, or energy storage molecules like glycogen. Amino acids come together to form proteins. Then proteins, along with lipids, might make a plasma membrane. Multiple proteins together can form a larger protein or a whole complex. Or you can have proteins with DNA that are made up of nucleotides to form chromatin, and then chromatin make the chromosomes in the nucleus and so forth. So it's like a structural hierarchy going from small to large. I kind of said it backwards. Just slides. And carbon has a unique role in life because you have the 4 lone electron pairs. You can form so many diverse compounds from carbon. You guys learned that from organic chemistry. You can have all kinds of different compounds. Huh?
Yeah, maybe bonds.
The what?
The complement, the double bonds.
Yeah. You can form double bonds. You can even form triple bonds. E groups. And these, like a double bond versus a triple bond versus a single bond, they all represent what? Different oxidation states of carbon. So. And then there's 30 elements that are essential to life. And I gotta remember Or did somebody say it? Somebody said it. That was nice. So, bulk elements are going to be hydrogen, nitrogen, potassium, calcium, phosphorus, sulfur, chlorine, oxygen, nitrogen, and of course carbon. So, and then we have metal ions that are, trace elements, a lot of trace elements that are required for metabolism. A lot of them play an important role in catalyzing reactions. Magnesium, chromium, molybdenum. They're about to start making that across the street, at least molybdenum 99. Manganese, iron, cobalt, we use to transport oxygen. It's also used to catalyze reactions, carry electrons. Cobalt, nickel, copper, zinc, selenium, and iodine. Common functional groups in biological molecules. Obviously, there's methyl groups we can add. We can have ether bonds. There's ethyl groups, 2 carbons, and so forth. Phenyl groups, where you have a phenyl ring. Acetyl groups is where you have an acetic acid attached to some R group. An ester, we get lots of esters between, an ester, you remember, is a bond between an alcohol and a carboxylic acid. What's 2 carboxylic acids? Are anhydrides. We'll see those. Even more than those, we see phosphoanhydrides. That's where you have an anhydride between 2 phosphoric acids, which is down here. And amido, carboxylate. So for the most part, carboxyl groups are going to be deprotonated. So they're the carboxylate groups. Carbonyls, you guys remember that term? So that's just a carbonyl is like oxygen with a double bond towards carbon. And then, so you can have a ketone or an aldehyde. Remember those terms. And an enol is where you have an ene and an alcohol. You ever heard of those keto enol shifts? You don't remember that? So these, like, if you had another carbon here with the 3 hydrogens, these will exist together in an equilibrium. It heavily favors the carbonyl, the ketone. But you'll get a keto-enol shift once in a while where you have the ene and the alcohol. So if you get this, these electrons come down, pick up this hydrogen, these electrons will drop down the other carbonyl there with the ketone. And that's a keto-enol shift. That's important when we're introducing mutations in DNA. Plus it'll be important for some of the metabolism that we see with glycolysis. So biological molecules often have multiple functional groups. So and this is an example, acetylcoenzyme A. I know you guys are familiar with that. You've had to memorize, the TCA cycle. Acetylcoenzyme A enters the TCA cycle and delivers the acetyl group. So you have an acetyl group that's in a thioester bond with the active part of coenzyme A, which is this thiol group. Then you also have a middle group, and this is all just basically a chain to the handle of coenzyme A. If you look at the structure of this is adenine. It's part, this is basically a nucleic acid, right? You have phosphate here, this is a phosphoanhydride, and this, with the 3 phosphates, this would be ATP, except this phosphate's not in the normal place where you think of ATP. But this, so proteins and enzymes will recognize coenzyme A, and this is the handle part that is able to bind to the proper binding site. And this is the active site of coenzyme. The function of molecules is strongly dependent on the 3-dimensional structure. You guys remember stereoisomers? So one stereoisomer, like for drugs, for example, some drugs have stereoisomers, and they actually sell them as the stereoisomer. So you're not taking the inactive form that can add toxicity because you don't get any benefit from it, but your body still has to metabolize it. Or sometimes they can inhibit the actual stereoisomer themselves. One example of that, I think, is Adderall, right? You guys familiar with Adderall? That's one of the stereoisomers meant that. It was Dex. It's the Dex infinity. So that they only sell one stereoisomer because the other stereoisomer doesn't have nearly the activity as the Dex. And it's going to add toxicity. There's no reason to have that. You got geometric isomers. We all know that. Cis versus trans. You guys familiar with retinol? You know what that is? Or retinol? Vitamin A. I think it's 11-cis retinol is the pigment that's used in your eyes to absorb light. When you're that vitamin A absorbs the light, you guys remember the cis form of a molecule is the higher energy form. You boost electrons around that double bond, it has an opportunity to spin, and it's going to spontaneously convert to the trans form, which is going to alter the shape of the protein that's grabbed onto it, and that sends a signal that ultimately you perceive as you absorb the light photon in it. Does that make sense? So geometric isomers are going to be important to the activity of retina. Enantiomers, mirror images, they have identical physical properties except with regard to polarized light, and they'll react identically with actual reagents, but they might not bind identically. And then diastereomers have different physical and chemical properties altogether. And here we go through some of the examples. Cis versus trans, malic acid versus fumaric acid. Their structures. This is old organic chemistry stuff. This is what I was talking about. 11-cis retinol, or vitamin A. It absorbs a light photon and it will automatically, spontaneously convert to all trans retinol. And that sends a signal to your brain saying you absorbed a light photon on that cone or rock. And enantiomers, mirror image versus each other. Old review stuff. Another example of enantiomers and diastereomers that you can actually taste or smell the difference is the difference between R-carbone and S-carbone. And this is the flavor you get from spearmint versus caraway seeds. Big difference in flavor, and it's just the orientation around this bond. So interactions between biomolecules are highly specific, and macromolecules will have unique binding pockets for the proteins, and other macromolecules have unique binding pockets that'll only allow certain molecules to fit in well and bind, and binding of chiral biomolecules are stereospecific, like I was talking about with the what's it called, atom. And this is an example showing a specific binding pocket on a particular protein. So organisms perform energy transductions to accomplish work, and so forth. And we're really running out. So I'm trying to work through this. So ATP is like the chemical currency in the cell. We know that. And if you look at this, this is a high energy molecule with phosphoanhydride bonds. And one of the reasons it carries so much energy is you have all of this negative charge all pent up close together. When you break this bond, you've released some negative charge from all of that, plus you've increased entropy. Right? You've made two molecules out of it. And ATP, though, it can be broken just by breaking this phosphate off, or you can make inorganic pyrophosphate come off. You'll see this a lot in the collaboration. So how do you speed up reactions? We already talked about this a little bit. Lower the activation energy. So one way, though, that we typically do in general chemistry is we raise the temperature to speed up the reaction. What does that do to you guys? It raises the average amount of kinetic energy those molecules have. So you guys familiar with the reaction coordinate? You have, free energy, gives free energy versus, you have your reactants, some kind of activation energy, and then products, right? The difference between here, this is your change in free energy here. reaction. Here to here is your energy evacuation. Sorry about that. My handwriting requires this. So, but when you raise the temperatures, that speeds up the reaction by lowering the activation energy also, because you've raised the average amount of energy that the reactants have. So it moves it up here. I should use a different color. Does that make sense? And that's how, and basically, this isn't any higher. You just made the difference smaller. You can also increase the concentration of reactants. We do that in biology too, but on a small scale. That's what having high, some of the catalysis comes from bringing things together, where basically they have a local high concentration and they're together and in the proper orientation for that reaction to occur. But when you increase the concentration, you're just increasing the likelihood that two of the molecules will react, because they'll have the right amount of energy and the right orientation. Change the chemical reaction by coupling to a fast one. This is universally used by living organisms. See a lot of that later. And we can lower the activation barrier by catalysis, and that's what enzymes do for the most part. They lower that activation. So unfavorable and favorable reactions. Synthesis of complex molecules and many other metabolic reactions requires energy. So that's inorganic. Reaction might be thermodynamically unfavorable, meaning it has this free energy is greater than zero. And they put this little hash here. Do you know what that means? Huh? Well, yeah, the degree sign or whatever you want to call it. But what does that mean in this? This is your standard state free energy change. Do you know what standard state conditions are? 1 atmosphere pressure. Yes. Standard temperature and pressure. And biochemistry does not occur at standard state conditions. You know, standard state conditions is also all the reactants are at 1 molar concentration. We don't have much of anything that's above 1 molar in our bodies. Maybe water is definitely more than 1. But so this is standard state conditions. And creating a order requires work and energy. Metabolic reactions might have too high energy barrier. And that's what this little plus is. This is the change in this is the free energy of activation. So that's greater than 0. Metabolite is kinetically stable. Breakdown of some metabolites releases significant amount of energy. That's exergonic. And such metabolites such as ATP, NADH, and ADPH can be synthesized using energy from sunlight and other fuels. So we create these by burning sugar and fat from the things we eat, and plants can make them. Their cellular concentration is far higher than their equilibrium concentration, so that's going to drive things forward, because we have much higher concentration than at equilibrium, so that moves the reaction for them to break down ATP and ADP. So energy coupling. Chemical coupling of exergonic and inorganic reaction allows otherwise unfavorable reactions to occur spontaneously. And one of the examples of this is the first step in glycolysis, we add a phosphate to glucose. So that's not going to occur spontaneously just with the phosphate, just adding it to glucose. We use ATP, we take the phosphate from this reaction. So we use ATP. We remove the phosphate from ATP to phosphate. We couple it to this reaction, so this has a positive free energy change, which isn't going to occur spontaneously. When we combine the two, which is basically coupling this reaction, the energy we get from this reaction, to making this reaction occur, it can actually be spontaneous. Does that make sense? So we use the energy we get from converting ATP to ADP, to convert glucose to glucose 6-phosphate.
How is that coupled? Like, do they just happen right next to each other, or are they?
No, actually, so this phosphate, rather than just being cleaved with water, is cleaved with the hydroxyl group on the sugar. So. And then a catalysis is a compound that increases the rate of the chemical reaction. Catalysis lowers the activation of free energy, and it does not alter the change in free energy of that reaction. So the catalysis is just going to speed up the reaction, whether its equilibrium is forward or backwards. So it doesn't change that free energy. Enzymatic catalysis offers acceleration under mild conditions, high specificity, so it'll only catalyze the reaction you want, rather than, you can use platinum and that will catalyze lots of different reactions. Or possibility for radiation also. So you can modify enzymes so that can either turn them off and turn them on. And, this is the figure that I drew here kind of